the Gordon Center for Integrative Science at the University of Chicago, this is the Pulse Podcast. I'm your host, Tanner Baldwin. The Pulse dives deep into the most interesting biotech stories and features world-class biotech leaders. This podcast is brought to you by the Harvard Biotech Club, a trainee-led organization that is on a mission to bridge the gap between academia and industry. Today's guest is Dr. Chuan He. Chuan is the John T. Wilson Distinguished Service Professor in the Departments of Chemistry and Biochemistry and Molecular Biology at the University of Chicago. He is also an investigator of the Howard Hughes Medical Institute, and his research sits at the intersection of chemistry, biology, and medicine. He is widely recognized as one of the pioneers of the field of epitranscriptomics. Chuan received his Bachelor's of Science in Chemistry from the University of Science and Technology of China in 1994. He earned his PhD in chemistry from MIT in 2000, and then completed postdoctoral training at Harvard before joining the University of Chicago faculty in 2002. Over the course of his career, Chuan has transformed our understanding of how chemical modifications on RNA regulate gene expression. His work showed that RNA methylation is reversible, a discovery that opened an entirely new area of biology and has had major implications for cancer, development, plant biology, and other fields. In recognition of these contributions, he was awarded the highly prestigious Wolf Prize in Chemistry, as well as the Tetrahedron Prize and the Falling Walls Science Breakthrough of the Year in Life Sciences Prize. We are thrilled to have Dr. He with us today. Chuan, thank you for joining us.
Well, it's great to see you again, Tanner.
Now, this is a special episode of The Pulse because we're here in the beautiful Gordon Center for Integrative Science on the campus of the University of Chicago. This is our first episode taking place outside the greater Boston area, and we feel really lucky to have you as a guest. It's a testament to how far the Pulse podcast has come in such a short time, thanks to the hard work of the Pulse team. But before we start discussing your exciting scientific accomplishments, as well as your future plans for translating that science into biotech advances, I'd love for you to start just by telling us a little bit about your background, specifically your upbringing, and what originally inspired your interest in science.
Yeah, well, first of all, Tanner, thanks for actually involving me into this program. Thank you. It's really a pleasure. That I get to ask that quite a bit. I grew up in the mountain areas in southwest of China. My parents graduated from college, and then back then in 60s, they were assigned to work for this I was in manufacturing industry deep in the mountains, because that was a time during Cultural Revolution. There were tensions between Soviet Union and China, so the Chinese broke many of their manufacturing into half. One half stayed in the city, the other half they moved into mountains just to back up whatever in the cities. So that's where my parents went. And it turned out many college graduates were assigned to work in these different units. So the average education level there is way above the rest of China. And I think there, growing up there, having the environment filled with sort of intellects and variety of diverse people from different background, right? That was stimulating. I think early on, you hear about Einstein, you hear about physics, Big Bang. We're talking about 70s, 80s, right? With limited exposure to the other part of the entertainment, right? So that kind of shaped my I think early days. I was like, I want to be a scientist, right? From there, it became sort of pretty much a straight line, and I went to college and was determined to come to U.S. because that's where the most exciting frontier science done, and came to get my PhD training at MIT. Did postdoc at Harvard, so I lived in Boston for 7 years, and I started my career in Chicago, and I stayed in Chicago since. So that's a simple, interesting, I guess, path. Yeah,
definitely. Yeah, it sounds like from an early age you were in a very intellectually stimulating environment. Were your parents academics? Did they have any sort of scientific affiliation?
Not really, but my dad is a physician, so he graduated from med school. My mom was a chemist, but I think the factory we were in were really manufacturing plants and other things. So my mom did not really practice her own science. It was a very bizarre system, I would say. People were not very happy. They were just basically packed there. But I did get exposed to all kinds of interesting, I would say, different people's different thoughts. When we were young, there were telescopes. We would look at the stars. This type of thing. You definitely get that exposure.
Yeah. Did you ever consider, following in your dad's footsteps and going to medical school, or did you always know you wanted to do science?
Well, the first thing my dad told me is don't be a physician. I think back then, physician in China back then was respected, but there's more service, and I think the young kids there always have this sort of desire to get into science and be the next Einstein or something, sure. Right? It's like every kid's dream, right?
Yeah, absolutely. And so you mentioned sort of being drawn to the States and the sort of cutting edge research that's done here, and of course you conducted research at MIT, at Harvard. Ultimately, you ended up at the University of Chicago. Is there a specific reason why you ended up here?
Well, I was actually towards the end of my first year postdoc, and then my PhD advisor just wrote me an email, said, go find a job. I was like, okay, if you think so, let me go find a job. And then I always joked the University of Chicago made a mistake. They hired me. And I ended up here, but I want to actually say, I said this a few times, I really regretted my decision because I should have stayed as a postdoc for actual 1 or 2 years. Because when I came out after like 22 months of postdoc, I don't feel like I was experienced enough in biochemistry, in biology, because my PhD training was pretty much in synthetic, inorganic chemistry, but my postdoc started really biochemistry, structural biology, a little bit of molecular biology. So I was not ready, but I was also lucky I was able to come to Chicago and grow in this wonderful environment.
Yeah, I can imagine that having a diverse background at times can be a strength. Have you found that's true, or you felt like maybe a few more years as a postdoc specializing in chemical biology would have been more productive?
I think it would be more productive. Back then, there are much fewer people sort of have cross-discipline experience. Unlike today, right? It's very common. We're talking about like 26 years ago, right? I think I did not receive enough biological training during my postdoc, just because it's short. There's a lot more I can learn from the lab. So when I studied at the University of Chicago, I really sort of went back to synthetic chemistry and played with structural biology, a little bit of microbiology. So it took me 5-6 years to get tenure. And only after my tenure, I sort of made the decision, it's time for me to switch. But also, I think in that 5-6 years, I've learned enough biology by working on microbiology, biochemistry in my own lab, and DNA damage repair, for instance. I gained enough confidence and then background to tackle bigger questions.
Yeah, absolutely. Whatever lack of experience there might have been at the outset, I think you've certainly been able to sort of make up for that and then some. Now you're at the University of Chicago, and at what point did you become interested in nucleic acid modifications? What point did you begin researching this question of RNA modifications? Right, these were known previously for many decades, but your work opened up this whole new idea that these were reversible, dynamic. Can you tell us a little bit about sort of how you got interested in that question?
Yeah, so when I did my postdoc work, I was working on DNA damage repair. So I basically picked Professor Greg Verdine's lab, learned all the DNA methylation repair. So I was working on ADA protein, and this is a transcriptional factor in E. coli, regulates ADA itself, ARC-A, ARC-B, ADA-B. So that was my postdoc work, and I become very interested in ARC-B because at the time they were computational analysis suggest that this is a metalloprotein, it's an iron protein, may perform oxidation. So I mean, with a metals in biology background, my PhD training was a synthetic inorganic chemist interested in mimic enzymes, particularly iron and copper enzymes. So I was drawn into this and wrote a job proposal to study DNA damage repair, particularly oxidative demethylation repair. At the time was something very new. So my lab spent the next, I would say, 5-6 years really adding different pieces into this very novel DNA damage repair mechanism. So we've been doing interesting, I would say, DNA modification chemistry and biology from 2002 to 2008. So 2008 was the time I got my tenure, and I already say this is actually true. Once the day I got tenure, I thought about it, and then I made the decisions to close many part of the lab. Pretty much almost shut down the entire program, but that takes time because you have students, postdocs, right? But I made the decision I was going to shut down pretty much all the programs. We've done very well, I would say, in structural biology of DNA damage repair, microbiology, metals, proteins, and some synthetic work, and looking for new areas of interest. And I was lucky. I talked to my colleague Tao Pan, who's an RNA biologist, and we kept talking almost every other week, and came up with several ideas. We thought about RNA imaging, and then we realized there's good people working on RNA imaging already, and then we thought about RNA modification. And the third topic we thought was actually SNO-RNA. Tao brought this up, and just a few years ago we started to work on SNO-RNA. But at the time, when I look at my expertise, and I realized chemical modifications on RNA is a very interesting subject. So that was the area I put efforts on. And we were, I would say, I think in science you also need to be lucky. And we were working on FTO, the protein, at the time because it was proposed to be a DNA damage repair protein involved in obesity, diabetes, and it took a couple years for us to realize that its real substrate is RNA methylation, and then the rest is the history.
Yeah. It sounds like, acquiring tenure in 2008 would you say that was a pivotal moment for the lab? On one hand, you're, shutting down certain focuses, but on the other hand, you maybe have more flexibility to take more risk, work on an area that fewer people are working on.
Yeah, I think there were two factors. Number one, of course, you got job security, right?
Yeah.
Yeah,
that's a big one.
That's a big one. That's important. You get that monkey off your back, right? The second part was actually back then they were mechanisms to support sort of ideas with no preliminary results. So we have this Eureka grant from NIH. It's $200,000 a year, Tao and I also had a CAC Foundation grant that was like $1 million for 5 years. When we're talking about almost 20 years ago, the money went much longer ways than today, right? So with that financial support, we were able to spend 3-4 years work on something, we don't know it's going to pan out or not.
Yeah. So a very long story short, it ends up it does pan out with this FTO discovery. I was wondering if you could maybe expand on that a little bit and why that was such a big discovery.
Right. I think this goes back to epigenetics, right? It goes back to complexity of human, right? We each have trillions of cells and 200 different cell type, depending on who you talk to. But we all came from a fertilized egg. What that means is the majority of our cells, the genomes, pretty much share the same sequence, right? That's 3 billion base pairs. So we're talking about 3 billion base pairs encode complexity of trillions of cells. And 200 different cells, but thousands of cell state. If you think about stem cell, progenitor cell, and then cells under stress, different state, right? What encodes this complexity, right? It has to come to certain modifications on DNA, histones, right? And on top of that, there's also RNA modifications. So before our work, we all know DNA modifications, histone modifications are very important to encode complexity of the genome, and then post-translational modification of proteins make millions of different forms of protein, right? That's where the complexity of life comes from. So people tend to pay less attention to RNA modifications. The thought back then was RNA modifications fine-tune RNA structure. Right? I think we came in, proposed in 2010, RNA modifications can have similar regulatory functions, right? And I think the discovery of FTO as a demethylase really showed that FTO is such an important protein or gene with very significant phenotypes. If you knock it out in mouse, it can be lethal. All right, surviving mice sort of a slower development. It's associated with energy, metabolisms, etc., etc. So everything suggests this is a regulator of diverse physiological pathways, and yet it's actually an enzyme removes a RNA modification, right? And I want to emphasize that the modification itself is also particularly interesting. This is N6-methyladenosine, right? This was discovered back in 1970s. And the most abundant modifications on mammalian messenger RNA, if you count, it's about 2-3 per messenger RNA. 40% of messenger RNAs could be modified, so the modified one carries 4-5 of this, right? But back in 70s, 80s, because of lack of molecular cloning, all the modern technologies we have, people don't really know what these modifications work on, right? These are low abundant RNA. And then later on, the fact that FTO reversed this, right, suggests the broad regulatory function, and later on we found this has profound impact to pretty much every biological process, and in the last 5-6 years, we further showed modification on chromatin-associated RNA that shapes transcription, etc., etc. So I would say this is a sort of a new way to look at the gene expression regulation, and this new layer of regulation seems to impact pretty much every biological process.
Yeah, it was definitely a profound discovery that really introduces a whole new paradigm that the sort of complexity, the diversity in regulation that is applied on the DNA level also applies to RNA. And with that FTO discovery, it introduced an idea that these modifications are highly dynamic and have very profound impacts on things like cancer and plant biology. And it was your work that sort of kicked off this whole journey. Can you tell us what it was like being there at the beginning, and then following this field as it's grown and matured, and maybe some of the challenges of sort of pioneering a field when there's not established ways of studying these things, not a lot is known. It was your work largely that built out a sort of repertoire of tools to study these modifications. So maybe if you could talk to us a little bit about some of the challenges of being a pioneer in a new field, and also, what sort of motivates you more? Is it the biological question, or is it developing the tools that could unlock unknown biological questions?
Yeah, these are very important questions, right? Very interesting questions. I think early on you get this excitement because you suddenly realize you open a door, right? There's everything you can explore, right?
Sure.
The challenges is which one you really hone on, right? Which one will have broad, long-lasting impact, right? We went to the M6A binding protein, the reader proteins, because we reasoned that by delineating the regulatory pathways, we can sort of describe to the community how these modifications impact fundamental RNA processing. And then people can take this to their own field of cancer, immune, right, stress response signaling, and see how this lay out. There are other challenges at the beginning. I think really because of me, because my training was not in RNA at all. So we had to learn everything. That was a lot of challenge. We did not know histories. We did not really know the literatures or not familiar. So that creates many challenges and sometimes maybe confusions with the field. But I think we handled it well. And end of the day, we were able to help build the field and really demonstrate the broad, profound impact of this regulatory pathway in diverse biological systems. Regarding the last question, I think you really need to look at the biology. But often the new biology is driven by new technology, right? We all know sequencing, mass spec imaging, right? These are the things that really promote sort of accelerates biology, but open new areas of biology. I would actually emphasize both. My lab is absolutely a method development lab, but then we often have our own biological questions. Because of that biological challenge, we go back and ask, can we develop methods to help us unlock this biological challenge?
Yeah. Do you think it's a particular advantage of yours to have sort of both of those under one roof? On one hand, you have members of your lab that are sort of expert in these new tools, but you also have those that are maybe more interested in focusing on the interesting new biology. And you have both sort of in one. How do you sort of see that playing out in your work and the collaborations between your lab members?
Definitely. As I said, sometimes you have a biological challenge and then you come back to develop methods, right? But there are also cases, it's just, I'm a chemist, right? And then I dressed like a biologist. But inside I'm a chemist and a biologist, I would say, you just have a very interesting idea you want to test out. You don't know what it's going to be used for, right? Like we developed this enzyme-modified Kisoxo. I thought it's cool to label single-stranded DNA, right? And then we developed Cas-seq, which can really map transcription. By the way, I think a lot of people should take a look at the Cas-seq. Kas-seq is really good for transcription mapping. And we could never imagine, that becomes sort of the key later on for us to study RNA interactions, which become a major part of the lab, right? So as you said, having both under one roof allows you to sort of think, I've got this method, right? I've got this scientific challenge. Maybe I can further modify and then use to help me solve this scientific challenge, right?
Absolutely. And you've had a lot of success at doing both of these, answering, interesting biological questions and also, developing the tools that are pushing this new field forward for others to use and make important breakthroughs with. And, deservedly you've won many prestigious prizes, including the Wolf Prize, which is often thought to be a sort of precursor to the Nobel Prize, the Falling Walls Prize, many others. And after having such an illustrious career as an academic, in recent years, you've turned some of your attention towards translating these really important and exciting scientific breakthroughs into potential therapeutics, into diagnostic tools. Could you tell us a little bit about some of those efforts and what you may be most excited about?
Yeah, I think in the last 10 years, we've shown that the RNA modification, the epitranscriptomics, the pathways regulate essential, important biological processes, and their misregulation affects human diseases. Right, in our hands, really affects, for instance, stem cell differentiation, development, proliferation, and also immune systems. We'd like to have our research add new knowledge, but on the other side, I also try to emphasize you want to impact the society, right? So
absolutely.
On that front, we spend a lot of efforts lately trying to really show these discoveries can impact a patient, right? We ourselves have been looking at cancer therapies, immunotherapies, diagnosis, as you said, and also plant biology, and there are other efforts in the community, right, in clinical trials targeting METTL3, targeting M6A binding proteins, erasers. My prediction is in the next 3-5 years, we're going to see really a burst of interesting clinical results that I hope will sort of validate our basic research.
Right. And your work sort of follows a similar trajectory to, the academic community as a whole, in a way that the barriers between academia and industry have sort of broken down in recent years. Can you tell us a little bit about your sort of philosophy for your students? If you have any maybe words of wisdom for individuals, trainees that are looking to, do really interesting and exciting science, but of course they also want their work to have that, maybe direct impact to patients' lives. Do you have any advice for people like that?
Yeah, I think first of all, when you receive academic training, right, you want to become an independent thinker, right? Mm-hmm. That's number one. Number two, always keep in mind, you have to ask yourself, where's my value? Where's my innovation, right? Mm-hmm. You bring in value because you either have an innovative approach, innovative idea, or you're capable of catalyze that, right? So these come from your academic training and your research, right? From there, I think you want to become open-minded and keep in mind research and the translation are two connected but a very different thing, right? The research is about uncover new knowledge, develop new tools. But translation is about directly solve medical problems, right? So the second part is more targeted, right? You have to show you can solve the real world problem. Otherwise, your program is going to die, right? Unlike the first one, as long as you're doing interesting science, you'll be supported. But the second one, not the case. If you don't have market value, you disappear, right? It's a very simple game. I think you want to have sort of your value from your training, your innovation. But when you go to the real world, you really need to ask yourself, what are the problems or questions you can solve that has real market value?
Yeah. Do you find it sort of challenging at times to balance, responsibilities that you might have relating to your work in industry, as an academic, as a mentor, very different priorities?
Yeah, that's a great question. We're setting up a therapeutic discovery institute here at the University of Chicago. We're known for basic discoveries. Now we're trying to make social impacts. We ask the exact same question. From my point of view, there needs to be a war in between, right? On one side is academic exercise, which is what we're supposed to do. That's innovation. Right, what's unique about you? Why you? Right, what can you bring that leads to new therapy? There, it's a free spirit innovation. On the other side is actually translation, and that the scope needs to be defined by the industry, not the professors, right? There's very few that fully understands academia versus industry, right? And that perhaps can go back and forth, right? You've got several in Boston areas, very good names everybody knows. But you have to really learn from failures, mistakes to understand, right? Again, it comes back to everybody doing their own job as a professor, as an academic researcher. You innovate new things, right? And in industry, you execute and have a clear goal, a clear exit strategy, right? Bring this thing eventually to the bed.
Yeah, absolutely. You definitely want some element of specialization. I think you're right. If there is too much blurring of the lines between academia and industry, there's also blurring of those different motives, right, that maybe shouldn't be blurred, a profit motive versus a motive maybe to pursue truth above everything else. So I think that's, very wise advice.
I also want to say, despite that, there's a trend of incubating biotechs directly from university labs. And
right.
I think that's going to be the future as well. Because we've got everything, right? Translation, we've got the entire machinery set up for translation, right?
Sure.
We have VCs, early stage, mid stage, we have farmers, right? So incubate something is not that difficult anymore, right? And then, I think the days you get $30, $40 million to start something completely new is also not financially feasible.
Sure.
So I think we're going to see a lot more incubations in a very flexible mode, right? Particularly because of federal funding has been challenging. Right? We're going to, I think, see a lot labs involved in early stage validation of ideas, early stage sort of incubation of new ideas, and that lab has to make the innovation, right, and then sort of deliver that innovation to the next stage before a real biotech takes over.
Right. Yeah.
I think we're going to see a lot of those.
Definitely. Yeah. Almost as if there is a wall, you want it to be semi-permeable to some degree, at least communication between
Exactly. That was set up because there's a two different mentality. However, there needs to be flow, and there's going to be a lot of flow. I think that wall is particularly important when we're going into the next era in which labs is going to incubate projects. And then the professors need to realize your research versus the real product is
definitely not always easy to know at the outset. A lot of these therapeutics, right, it's something like 10% ultimately get FDA approval. So,
or 5%.
Yeah, maybe less. Is it safe to say that maybe you see these sort of issues with federal funding potentially leading into a new paradigm in the way that academia relates to industry, with industry picking up more of the slack of the basic research, the foundational research? Is that something that you see persisting and being good for the future of science and industry?
Yeah, I think I've thought about this years ago. We're so heavily rely on federal funding, right? And federal funding has played such a transformative way of technology science advance in U.S. because basic research is really the key to everything. So I think the federal grants should continue to play that essential role. And I think in the future, I picture three sort of funding sources: enterprise, or community. Federal grant has to be strong, right? That's sort of the foundation. The philanthropy foundations, gifts, right? I think that kicked in a lot, like Howard Hughes and different foundations. And I think, like Scripps, like these other institutes, we also need to have a portion of our labs or a portion of our own research perhaps be directly connected to industry, right? It just depends. Their PIs just interested in basic research, perfect. But we emphasize heavily on that. In the PIs interested to bridge basic to translation, right? There's nothing wrong with that,
Sure.
and that will really show the impact of basic research, right? So I think everywhere we're going to see the build up of that type of hires and that type of labs, and also in some of our own labs, we're going to have some projects directly working, hopefully, with industry and solve major questions there.
You know, you raise a good point that we've faced recently some challenges with federal funding, but there is, you know, an argument to be made that at the very least a underutilized resource with the potential collaborations between academics and, you know, industry resources. So maybe these sort of difficulties that we're having now are an opportunity to reevaluate, the sort of underpinnings of how science is driven.
Yeah.
Yeah. So ultimately, you know, you've had this, you know, amazing career as an academic. Recent years, you've made moves in industry as well. If you could sort of go back and chart your course anew with hindsight, would you do anything differently?
I would like to learn the industry part in a more sort of I was rushed into this in two cases. I don't want to name, almost against my own will, and neither of those worked out well. So I'd like to actually have, I think, more naturally engage this. I think our younger generation of faculties or PIs are different, right? They grew up in this era. They naturally started to embrace industry, right? There are opportunities for them. They talk to VCs. They watch how companies been set up, right? As I said, this is almost like research. If you don't fail, you don't learn. So I'd like to actually, if I had the chance, sit in some advisory board or have a direct interaction with industry, but in a minor role at the beginning to learn, experience this a few years before I fully get immersed into something.
I think that's good advice. Just as you said, as the sort of scientific enterprise becomes more interdisciplinary, it's important for students to sort of be keeping up with those changing times and to some degree having a sort of diverse training experience. So just to wrap up, I'd like to ask some more lighthearted questions. When you're not doing science, what do you like to do?
Well, I like travel a lot. My wife and I, we travel the world. We take our kids and, we went to Iceland, Norway, hiking, right. Just experience the world. And, it's the same sort of a curiosity in science, but also curiosity of the other part of the world, the new things, the culture, how people live there. Absolutely.
Yeah. If you couldn't be an academic, what would you do for a living?
Not going to play soccer, as you know. Tanner is a great soccer player. No, I always joke that, my ideal career would be a coach for a football team. Right?
Okay.
Yeah, and I'd love to do that.
Yeah. Awesome. Do you watch a lot of soccer?
I watch soccer, but I watch a lot more football, American football.
Okay.
Yeah. No, that's my sport.
I see.
Yes.
Do you have a favorite team?
I know. I'm sorry to say I've been a Patriots fan for 30 years.
Okay. Great. Yeah, maybe the Chicago Bears can come in as a close second.
Yes. Yeah. I bike down to watch their games, but they're going to have a new stadium, so hopefully I can still go.
That's great. Is there any a book or any piece of media that you would recommend students listen to or read, or something that had a profound impact on your career or journey?
Not particularly, actually. I'm sorry to say that, but I do have advice. I do advise undergrads to get into lab as early as they can if they're really interested. Have a hands-on experience is better than taking 100 different classes.
Sure. Yeah, there's really no book could ever replace being in the lab.
Exactly. And then also, if you want to do translational work, just spend an internship in a company, right? Or work on a project, right? Again, hands-on experience, the real-world experience is so important.
Yeah, absolutely. Well, thank you for taking the time to sit and chat with us. Really appreciated it.
Thanks, Tanner. It's great to have you come back, and great to see you again.
Thank you.
Yeah, good luck.
This is the end of this episode for the Harvard Griffin Jesus Biotech Club, the Pulse podcast. I'd like to thank Tanner Baldwin for co-hosting this episode. Subscribe to our podcast on Apple Podcasts, Spotify, YouTube, Substack. For more resources, please head over to our website, www.thepulsepodcast.org. Please stay tuned for our next episode. Thank you for listening.